Two trait crosses, what we're going to be talking about today. Here are your learning goals and success criteria. Take a couple seconds to read them. Okay, before we get into two trait crosses, let's do a little bit of a one trait practice. Here are two scenarios that I want you to try out on your own right now. So pause the video and try it out and press play when you're ready to take a look at the answers. So I highly recommend that you guys try these out, actually draw out the Punnett squares and figure out what the percentages are. Because if you're just looking at the answers and corresponding them to the questions, it's not really going to help you learn how to do this. And this is the very basics of what we're going to be doing over the next few lessons. And it gets more and more complex. So if we're able to master this, then we can move on to two trait crosses, that what we're going to be doing today. All right, so make sure you try these out. If you don't get it, ask the questions on the stream or in email, or find me during office hours. But we have to get this in order to get to the next section, or else you're going to get very confused at how you're going to do two trait crosses, how you're going to get the gametes. Okay, so we have to know how to do everything that's on this practice first. All right, so two trait crosses. After determining one trait inheritance patterns in pea plants, Mendel went for a second set of experiments. Okay, so he wanted to know if inheriting one trait has an impact on another trait. What he did was he started with plants that were true bred for two traits. So now we went from one trait to two traits, and where each parent differed in both of the traits. So this is called a dihybrid cross. And dihybrid cross is a cross of two individuals that differ in two traits due to two different genes, and that's why we call it two trait crosses. We're not just looking at whether or not it's round or wrinkled. We're not just looking at whether or not it's green or yellow anymore. We're actually combining the two traits. So the trait now we're looking at are color and whether or not the skin is round or wrinkled. So in Mendel's experiments, he crossed two true breeding plants. He crossed yellow round seeds together with true breeding plants that were producing green wrinkled seeds. He discovered that all the F1 generation plants displayed dominant forms of each trait. They all had yellow round seeds. And this result is identical to the one trait crosses, and after letting the F1 generation plants self-fertilize, the recessive forms appear in F2 generations, just like what happened in one trait crosses. So as you can see here, P generation, we have our yellow round peas fertilized with the green wrinkled peas. The first generation that showed up were all yellow and round, and he took all the yellow and round, fertilized them again, and then he got a mixture of all of the different types that showed up. Okay, so some of them showed up as yellow round, some of them showed up as green round, some of them showed up as green wrinkled, some of them showed up as yellow wrinkled. So in F2 generation, the ratio of plants with yellow round to yellow wrinkled seeds to green round to green wrinkled seeds was about 315 to 101 to 108 to 32. So if you round it to the closest number, you're going to get about 9331. What happens here is Mendel kept doing the same crosses over and over again, and no matter which trait he followed, as long as he's doing two traits, he got the same ratio of 9331. This means for every one offspring with recessive forms of both traits, as you can see here, it's recessive forms for both traits, there are going to be three offspring with one recessive form. So here's a recessive form, and one dominant form of the second trait. So here's our other trait, which is going to be dominant form. We don't know what it's going to be in genotypically, so it could be homozygous dominant, or it could be heterozygous. We don't know, so we're going to put an underline here just to state that it's going to show up as the dominant form, but we don't know what it is genotypically. It's also going to have three offspring with the opposite combination of what we just saw. So you're going to have our R's going to be shown as dominant. We don't know which genotype it's going to be. And we're also going to have the recessive form for color show up there. And then we're going to have 9 dominant forms of both traits that are going to show up. And that dominant form could be either heterozygous or homozygous dominant. So a Punnett square can actually model the segregation of the gametes for dihybrid cross experiments. In F2 generation, each of the parents can produce 4 possible gametes, since each of the allele for color could be combined with each allele for shape. So in 4 gametes from each parent combined, 16 possible genotypes are produced. So if we take a look here, they will produce the same genotype for F1 generation. And if we take out the gametes for the F1 generation, since they're the same, they have 4 different variations of their gametes, and all of them have equal chances. So now we're going to put them into our 4 by 4 because it's 4 gametes each. So it's going to be a 4 by 4 Punnett square. And here are our genotypic and phenotypic ratios that showed up. So Mendel called this the law of independent assortment, where the gamete, during gamete formation, 2 alleles for one gene segregate, or they will assort independently of the alleles for the other gene. So it doesn't matter what the different traits we're looking at. As long as they're different traits, those alleles are going to separate. So this means that color and whether or not it's round has nothing to do with each other. They will segregate on their own, but because we're looking at 2 traits, both traits will have to be accounted for. So here's our first sample problem. Actually, I'm only going to have one sample problem for us today, and then I'll give you your homework after that. So in wolves, the allele for brown eyes is dominant to the allele for blue eyes. So allele for brown eyes is dominant to the allele for blue eyes. And we have a different, because we have 2 traits. So we have allele for gray coat is dominant over black coat. So now we're going to predict the genotypes and phenotypes of the potential offspring of a blue-eyed gray wolf that had a black-coated mother and a wolf that was heterozygous for both traits. Okay, it's going to sound very wordy, very, very confusing, but we will take a look at it one at a time. So let's take a look first. What are our letters that we're going to be using to represent all of these stuff? So let's take a look at eyes. So eyes, it says brown is dominant here. Brown is dominant, so we're going to use uppercase B for brown, lowercase b for blue. Coat is your second one, and gray is dominant, so we're going to use G for gray, and then lowercase g for black. All right, now that we have this information, we will need to, because we have to cross them and we have to know what exactly we're looking at. So we have the first wolf, wolf 1, which is blue-eyed. So it says here, blue eyes, gray wolf. So if we look at blue, blue is recessive. So that means the eyes show up recessive, means that it has to be homozygous recessive. However, the gray wolf, the gray coat, it shows up as gray, but we don't know whether or not it is going to be heterozygous or homozygous dominant, so I'm going to put an underline here for now. Our next clue that we have is that this wolf that we're just looking at here, this wolf here, it has a black-coated mother. So for the black-coated mother, for it to show up as black coat, she has to have homozygous recessive. And she's going to pass one of those alleles to the offspring. So that means this has to be a lowercase g. So this is going to be a heterozygous for coat. Now let's look at our second wolf, which is heterozygous for both traits. So heterozygous for both traits, that one's easy. We're just going to put it into heterozygous here, and that's what we have. So before we can do this in the Punnett square, we have to segregate all of these gametes. And let's just write these down, what their gametes are, before we can actually put them into the Punnett square. So the gametes here, so remember each of the lowercase b's in the first wolf have to go with the other g's that it has, right? Because it segregates separately. Now, if it repeats, we can write it, we can choose not to write it. It's going to be the same. It doesn't affect our results at the end. So I'm just going to leave that out because it repeats. So there's only 2 possible gametes for wolf 1, and wolf 2 has 4. And I did another mistake here. It should be that and BG. Okay, so these are the gametes for wolf number 2, and these are the gametes for wolf number 1. So if I put them into our Punnett square, I can actually do a 2 by 4 Punnett square. You could do a 4 by 4 and repeat these gametes if you want, but the results are going to be the same. Okay, so here are results If you take a look at all the genotype that's here, I've put all the ratios down here as well. And if they've repeated, I actually simplified them into 1/4 instead of 2/8 because it's the same thing. And then from our genotypes, we can figure out what our phenotypes are, okay? So that's your answer right here. If you haven't tried to do this, please try it and figure out how to do the Punnett square because now it's a little bit more complex than what we did before. Okay, so this lesson is going to be, it should take around an hour to do the work. So I'm not going to post anything for tomorrow, but I want you to take a look at this. Try the questions out, complete problem set C. There are some homework pages that I'm going to post onto the Google Classroom as well. Do all the practice and take up your answers for problem sets as well, okay? And I will see you guys next time.